Alright students, welcome back. We are going to learn about meiosis today. So I'm just gonna read the textbook to make sure that we covered what you need to cover for your syllabus. And then I'll give you an example which I did myself here. I'm gonna explain to you how things move using this thing. Okay, meiosis is the process of cell division that occurs in reproductive organs to produce gametes that contain half the number of chromosomes, haploid, of the parent cells, which are diploid. So meiosis occurs in the testes for male and ovaries for female for animals and humans. Okay. Meiosis forms gametes through the process of gametogenesis and ensures that the diploid chromosome number of organisms that carry out sex reproduction is always maintained from one generation to the next. Meiosis also produces genetic variation in the same species. Okay, it is divided into two stages of cell division, that is meiosis 1 and meiosis 2. Okay, later on you'll see that the only difference between mitosis and meiosis is this part. Okay, meiosis 2 is exactly the same as mitosis. So same goes with meiosis and mitosis. They both have prophase, metaphase, anaphase, and telophase. I hope by now that you have memorized these phases in mitosis, M phase, or meiosis, M phase also. Alright, meiosis 1. Okay, let's start with prophase 1. Alright, during prophase 1, chromatin shortens, thickens, and forms visible chromosomes. The pairing of homologous chromosomes, which is the process of synapsis, remember, the pairing of chromosome, forms a bivalent. Okay, the bivalent is this one, this whole thing, or known as a tetrad, that is 4 chromatids for each homologous chromosome. Actually, 2 chromatids for each homologous chromosome. Okay, the crossing over process, that is an exchange of genetic material between non-identical chromatids, takes place during prophase 1. Crossing over produces a combination of genes that are new in chromosomes. So here, recombination, crossing over, you get new combination of genes in one chromosome. Okay, the point where the chromatids cross over is called chiasma. At the end of prophase 1, the nucleus membrane and nucleoli will start to disappear. This is the same as prophase in mitosis. Okay, spindle fibers are formed among the centrioles, also the same with mitosis. Okay, you can see right here the nuclear membrane starts to disintegrate. Okay, and then you can see the pairing right there between 2 non-identical chromatids. Okay, and then you can also see the spindle fiber starts to form between the 2 pairs of centriole. Oh, bivalent. Okay, this, oops. So there's 2 bivalents right here. So one is for this homologous chromosome and another one for this one. So 2 bivalents or 2 tetrads. Okay, so this cell right here actually has 4 chromosomes. Humans have 46 chromosomes, by the way. Imagine this, but there's 46 of them in one cell of your body. Each cell's in your body. That's amazing. Next up, metaphase 1. You can see the picture right here. Pretty similar to metaphase in mitosis, but instead of all of them align in one single line, the homologous chromosomes are gonna align at the equatorial plane side by side. So this one is gonna align next to this one this one is going to align next to that one because they are homologous. The homologous chromosomes are arranged at the equatorial plane. One chromosome from each pair of the homologous chromosome is tied to the spindle fibers from one pole cell, and its homologous is tied to the spindle fibers from the opposite pole cell. Okay, this part right here simply means that the spindle fiber from this one, okay, is going to pull the centromere from just one homologous chromosome. Okay, this one is going to pull this homologous chromosome. Okay, so it's not so in if you remember in mitosis, okay, one centriole, one pair, okay, it's going to combine with one side of the centromere and the other another side of the centromere. So these two will separate later in anaphase, but as you will see after this, okay, instead of the chromatids separate in anaphase for meiosis, the homologous chromosomes separate. And there you have it. Okay, like I said before, okay, so the homologous chromosomes are separated and pulled to opposite poles. Okay, the sister chromatids are still tied to the centromere. You can see right there. Okay, they are not separated yet. Okay, they will separate, but not yet. Okay, so in anaphase 1, the homologous chromosomes separate. The spindle fibers contract and cause each homologous chromosome to separate from its homologous pair and be pulled to the opposite poles. Each chromosome is still made up of a pair of sister chromatids tied to a centromere and move as one unit. So this whole thing moves as one unit. This one thing moves as one unit. And the last phase in meiosis 1 is telophase 1. So during telophase 1, the chromosomes arrive at the opposite pole cells. Each polar cell contains a number of haploid chromosomes that are made up of one set of chromosomes only. The spindle fibers will then disappear. Nucleoli, which is plural for nucleolus, will reappear and the nuclear membrane is formed. Okay, what it means by one set is that, so for each polar cell, okay, one polar cell, this one has one set, this one has one set. Whereas if you remember in mitosis, okay, when they separate right here, okay, during telophase in mitosis, this one has 1, 2, 3, 4. This one has 4. So in mitosis, each polar cells have two pairs for one cell. But in meiosis, especially meiosis 1, telophase 1, each polar cell has one set. Okay. The implication of this is that the number of chromosomes are half from the original one. So in the original cell, we have 1, 2, 3, 4 chromosomes. But at the end of telophase 1, we have 2 cells, but instead of having 4 in each cell, we have 2. 1, 2, 1, 2. So 2 is a half of 4. In mitosis, if you remember, prophase, it starts with 1, 2, 3, 4 chromosomes, but at the end, during telophase, it has also 4 chromosomes. Telophase 1 is then succeeded by cytokinesis process that produces 2 daughter cells. Both daughter cells produced are in the haploid condition. The interphase for meiosis 1 is usually short and the DNA does not replicate. So it's already replicated. After cytokinesis, the cell separates into 2. It usually will go directly into the next phase. Alright students, this is very important. Now, at the end of meiosis 1, you see, well, the cells already have the number of chromosomes. but you can still see the chromatids, the sister chromatids, are still not separated. Now this is a problem because in order for cells to work, they only need to have one copy, which means that it cannot have two sister chromatids in one cell. Okay, so these two have to be separated for the cell to work properly, and that is the reason why we have another step, which is meiosis 2, to make sure that each cells do not have sister chromatids, two sister chromatids at the end of cell division. Okay? Right. Before I start to explain about meiosis 2, let me emphasize that this whole thing is happening at the same time. Okay, remember from meiosis 1, okay, we get two daughter cells. Okay, now these two, each one is going to undergo meiosis 2. Okay, so one cell from meiosis 1 is going to undergo meiosis 2, and another cell from meiosis 1 is also going to undergo meiosis 2. Now, right from the get-go, you can see from this diagram right here, okay, it is basically mitosis, okay, but occurring in meiosis. Okay, let's just go through each phases one by one, and you can see why I said that these two are basically the same. So in meiosis 2, prophase 2, the nucleolus and the nuclear membrane disappear. Okay, nucleoli is a plural because we have two cells undergoing prophase 2 at the same time. Okay, the nucleoli and the nuclear membranes disappear. Each chromosome is made up of sister chromatids that are joined at the centromere. You can see that. The spindle fibers start to form in both daughter cells. Okay, so the spindle fibers right here are starting to form, and then those things are gonna pull the sister chromatids apart later. Oops, battery is almost out. The next phase is metaphase 2. So in metaphase 2, chromosomes are arranged at random on the equatorial plane for each daughter cell, okay, in one single line. Each chromatid is tied to the spindle fibers at the centromere from both poles. Okay, so one pole has the spindle fiber pulling one side and another pulling the spindle fiber from one side. Okay, so it's not just one, but it's two. If it's one, then the whole thing is going to be pulled to one side. But because we have two, it's gonna pull those sister chromatids apart. Okay, now this might not be important now, but it will be important later on, maybe next year, I don't know. But there is a reason why over here it says at random. Okay, now this keyword is very important when you learn about variation of cells. I'm not sure, probably next year because I don't see it in your syllabus this year. Okay, so be sure to put chromosomes are arranged at random on the equatorial plane for each daughter cell. Okay, 1, 2. Anaphase 2, sister chromatid centromere starts to separate. Okay, the sister chromatid pair separates and moves toward the opposite poles led by the centromere. Each chromatid at this stage is known as a chromosome. Okay, so this is one chromosome, one chromosome. When they separate, okay, you don't call them chromatids anymore. Okay, chromatids, you call them when they are combined like this. Okay, when they separate, each one of them is considered a chromosome. So the difference between anaphase 2 and anaphase 1 is during anaphase 2, sister chromatids separate. Okay, but in anaphase 1, homologous chromosomes separate. Last but not least, we have telophase 2. Okay, this is telophase 2. So chromosomes arrive at the pole of the cell, spindle fibers disappear, the nuclear membrane and nucleoli are reconstructed. The number of chromosomes for each daughter cell is half the number of parent chromosomes. Telophase 2 ends with a process of cytokinesis that produces 4 daughter cells that are haploid. Each haploid cell contains half the number of parent cell chromosomes. The genetic content is also different from the diploid parent cell. The haploid cells develop into gametes. Okay, let's go through this one by one. Okay, chromosomes arrive at the pole of the cell. Okay, they arrive at one pole. Spindle fibers disappear, the nuclear membrane and the nucleoli are reconstructed. Okay, spindle fibers gone, and then the nuclear membrane starts to form. The number of chromosomes for each daughter cell is half the number of parent chromosomes. Okay, this one, so we have 1, 2, 3, 4. 4 daughter cells for meiosis. Okay, they are half the number of parent chromosomes. So let's see here, we have 1, 2, 1, 2, 1, 2, 1, 2. So each cells have 2 chromosomes. Okay, all came from one parent cell that has 1, 2, 3, 4. 4 chromosomes. And telophase 2 ends with the process of cytokinesis. Cytokinesis is the division of cytoplasm over there. That produces 4 daughter cells that are haploid. Haploid because this one is half the number of chromosomes in the parent. This is diploid. This is haploid. The genetic content is also different from the diploid parent cell. Okay, this part right here, the genetic content is also different from the diploid parent cell. Happens, you can see right here. Okay, the contents are different. This one has, this chromosome particularly is different from the parent chromosome. Okay, this one is also different. That's why it says genetic content is different. The haploid cells develop into gametes. Okay, so each of these cells will, so in males, it's gonna develop into sperm cells, and in females, one of them is going to develop into an ovum, and the other 3 is going to die. Alright folks, that's it from me. That is meiosis. There's a lot of things happening right here. It will take time for you to digest every single information that I just spew out to you. If you have any questions, please do not hesitate to contact your teachers first who taught you in class. So for me, 4AF and 4AG, you can text me personally on WhatsApp, and the others can do the same thing to Miss Aini and Mr. Sertan. Okay, until then, I'll hear from you guys soon. Bye. Oh wait, before I forget, so you see this picture right here? Okay, remember when I said this one is the same thing as mitosis, the flow right there? Okay, let's go to mitosis. Okay, prophase, metaphase, anaphase, and telophase. Okay, they start with 4 chromosomes. They end up 4 chromosomes. Okay, these ones, they start with 2. They end up with also 2.